Hello, my name is Rakesh, and today I'll be talking about the popular sports supplement creatine. Creatine is one of the most widely researched and most widely available sports supplements. This is by no means an exhaustive dive into the effects of creatine, but I will be focusing on some of the basic biochemical mechanisms of creatine both in the body and out, as well as some of the potential consequences of these reactions as discussed in various recent research. Additionally, while I will be taking a look at this research, I will also be discussing some of the ways that creatine may be accidentally misused by the average person due to a lack of proper advice on supplementation. So, let's begin. Before we get into the specific mechanisms of creatine in the body, it's important to understand what exactly creatine is. Creatine monohydrate is a basic amino acid that is naturally produced in small amounts by the body, but it's also a popular sports supplement. It is also found naturally in our diets in trace amounts in legumes, such as beans, but it's found in its highest natural concentrations in the lean skeletal muscle of animal meat, since animals produce creatine in their cells just like we do. The best sources of creatine are lean fish, like tuna or red herring, which can contain up to 1.1 grams per 100 grams of lean weight, and red meat such as beef, which can contain up to 0.9 grams per 100 grams of lean weight. As you can see, even when consumed in its highest natural concentrations, creatine is a relatively hard compound to consume enough of. The average adult can store up to 0.6 grams of creatine per kilogram of skeletal muscle, and so to obtain or even maintain the optimal levels of creatine would require an individual to consume kilograms of meat daily. So it is typically consumed as a sports supplement for high-level athletes that want to maximize the amount of creatine stored in their cells. So the question is, why would an individual want to maximize their creatine levels? Well, it's because creatine plays an important role in muscle contraction. While muscle contraction is a complex mechanism with many different compounds and pathways involved, the two most important factors are electrolyte levels and the amount of phosphorylated ADP or ATP present in your muscle cells. Electrolytes act as chemical signaling molecules that are used by your nervous system to signal specific muscle groups and or fibers to engage in contraction, and the basic mechanism of converting ATP into ADP creates free energy, which is coupled with the reactions used to contract and relax muscle fibers. Typically, it is very easy to maintain proper electrolyte levels through a proper diet and or the use of sports drinks. However, you cannot directly increase the amount of ADP or ATP in your cells because any ATP or ADP that you consume gets broken down by your digestive system. Additionally, rephosphorylating ADP into ATP requires a lot of time and energy. So, once your muscles have used their available ATP, does this mean that they can no longer contract? Well, this is where creatine monohydrate comes into play. Creatine, for reasons I'll discuss in the next slide, is really good at holding on to extra phosphate groups. This means that the creatine can quickly donate these extra phosphate groups to ADP, therefore quickly rephosphorylating it and allowing more muscle contractions to occur. This will allow an individual to either recruit a higher number of muscle contractions at once, which is beneficial for strength sports such as weightlifting, or for an individual to maintain a high level of muscle contractions for a longer period of time, which is beneficial for endurance athletes such as runners and swimmers. Additionally, as an aside, creatine is also used in other cells throughout the body, such as the brain, to quickly replenish ATP. A study from 2018 has shown that creatine supplementation can improve cognitive function as the replenishment of ATP allows neurons to maintain their ion gradients needed to generate and fire electrical impulses. Also, creatine has a very short half-life in solution, which is something that will be important later on. So now let's dive into the chemistry of creatine. Right here, we have a molecule of creatine monohydrate. As you can see, it's made up of a carboxyl group on the left, 4 carbons, 1 primary amine, 1 tertiary amine, and 1 imine. Because of its molecular structure, creatine is highly polar. The carboxyl group has a very large negative partial charge, and the nitrous end has a positive partial charge. In cells, the creatine is phosphorylated as shown by the reaction here on the right. This process occurs in the naturally occurring enzyme called creatine kinase, which is shown on the left. A phosphate group is taken from an ATP and replaces a hydrogen on the amine. This is an example of a regioselective substitution reaction, as the phosphorylation of the amine has a higher activity than the phosphorylation of the primary amine. You might be thinking, if ATP spontaneously donates a phosphate group to create creatine monohydrate, then how does a phosphorylated creatine donate its phosphate group to ADP? Shouldn't this reverse reaction be non-spontaneous? Well, as you can see on the left, creatine kinase has 2 different binding sites. And these 2 binding sites can actually be altered based off the levels of ATP, phosphocreatine, and ADP in the cells. So, That means that when there are low levels of ATP and high levels of phosphocreatine, the binding sites of creatine kinase are modified to allow the attachment of ADP and phosphorylated creatine to catalyze the reverse reaction, quickly turning ADP into ATP when phosphocreatine levels are high. Alright, so if increasing creatine in our cells is beneficial, then how come we can't store more than 0.6 grams per kilogram of skeletal muscle? Also, if creatine phosphorylation is reversible, then why do we need to keep consuming creatine? Various research has indicated that excess creatine spontaneously converts into a cyclic waste product known as creatinine, as shown on the slide here. Creatinine is formed when the carboxyl group is spontaneously removed from creatine monohydrate, or when the creatine phosphate is dephosphorylated without the presence of creatine kinase. These two reactions are an example of the dehydration reaction, as both of the reactions involve the removal of an H2O. Therefore, creatine levels naturally decrease over time, and excess creatine kinase degrades when it is not protected by the creatine kinase enzyme. Creatinine is considered a waste product, since it cannot be used in phosphorylation. Therefore, it is removed from the cells into the bloodstream, where it is then filtered by the kidneys. So, now that we know the general mechanism of creatine and how it can sustain high levels of ATP in our cells, let's talk about how creatine actually gets to your cells. So of course, the first step is to mix creatine in a liquid. Now as I've said before, creatine has a relatively short half-life of only 12 hours before it will spontaneously degrade into creatinine. This is where we run into our first issue. Many athletes prepare their meals ahead of time, and some may even consider doing so for their sports drinks. However, premixing creatine will cause much of it to degrade into creatinine before it is even consumed. Additionally, creatine degrades into creatinine much faster at higher temperatures. This is because the dehydration reaction is more spontaneous as you increase the temperature. Therefore, the improper storage of creatine at high temperatures, or more importantly, the consumption of creatine in hot water or in cooked foods, may reduce the amount of creatine that reaches the cells. Creatine's half-life is also greatly reduced in the presence of weak acids or bases. This is a very common problem, since many creatine plus pre-workout or electrolyte supplements that are sold over the counter will become weakly acidic when mixed with water. Taking this popular supplement for example, the citrates that are present in the ingredients will become citric acid when they are added to water, which will reduce the pH of the mixed drink. Adding creatine to sports drinks such as Gatorade also has the same effect, since many sports drinks are slightly acidic. These properties are also problematic since lowering the pH and increasing the temperature naturally increases the solubility of creatine. This could lead to an individual unknowingly degrading the creatine in an effort to make it more soluble. After the creatine is ingested, it is then brought to the stomach. One might assume that the degradation may worsen because the pH in the stomach is low, but that is not the case. Since stomach acid contains strong acids such as hydrochloric acid, studies have actually shown that it does not cause the degradation of creatine to drastically increase, rather it may have the opposite effect, stabilizing the creatine. However, supplements such as the ones shown on the slide may also have a slight buffering effect, increasing the pH just enough to cause the creatine to continue to degrade, since salts such as magnesium are weakly basic. Anyways, after passing through the stomach, creatine is then absorbed into the bloodstream from the small intestine, from where it is delivered to the cells. Now, I will be going over what is potentially the most dangerous consequence of improper creatine supplementation. It has been shown that microorganisms that contain the enzyme creatinase break down creatine into sarcosine and urea through glycolysis, which is the breakdown of an organic compound in the presence of water, as shown by the second step in the mechanism on the right. The sarcosine is then further broken down into glycine, formaldehyde, and hydrogen peroxide through the oxidation reaction by the enzyme sarcosine oxidase. Both of these enzymes are present in humans, meaning that excess creatine in the bloodstream may be broken down by this mechanism. Formaldehyde has been shown to cause problems in DNA replication, as well as a general degradation of DNA. So, does this potential mechanism actually play out in humans? While there is no direct research to answer this question, there are some studies that both potentially support and refute this claim. To start, a study done in the year 2000 administered high doses of creatine in mice, where it was in fact shown to increase formaldehyde levels in the blood, as well as cause kidney damage. However, a later study done in 2008 tested a more reasonable dose of creatine in human participants and found that taking the recommended levels of creatine does not cause any significant increases in the formaldehyde in the bloodstream or urine. However, an additional study done in 2005 administered significantly higher doses of creatine to participants. These young men who had previously not taken creatine were given 21 grams of creatine daily for 2 weeks, and this led to their formaldehyde levels spiking, increasing nearly 7-fold. However, no damage to renal vessels was found, meaning that there was likely no damage done to the kidneys. What we can take away from this data is that while proper use of creatine is not dangerous, higher doses of creatine may be. Many online influencers advise individuals to take larger than recommended doses of creatine when they first start taking the supplement, which is a practice known as the loading phase. This could be considered dangerous since the body will likely convert some of the excess creatine into formaldehyde if it is not properly absorbed by the cells. In conclusion, the lack of information and amount of misinformation readily available online can lead to the improper use of creatine. Creatine should not be consumed in acidic drinks such as sports drinks, and ideally, it should be consumed in room temperature water, since creatine that is at any higher of a temperature will denature into creatine much faster, and creatine will not dissolve properly in colder water. Additionally, the popular advice to use loading phases when starting to take creatine may be dangerous, as it increases formaldehyde levels in the blood in the short term. Furthermore, taking above the recommended dosage of creatine may be considered detrimental in the long term, as formaldehyde levels will remain high. Most importantly, however, more studies need to be done in humans in order to further understand the mechanisms of converting creatine into formaldehyde. Additionally, more studies should also be conducted on the other claimed negative side effects of creatine, such as the increase in male pattern baldness and increased blood pressure. Thank you.